Video of cell isolation experiment for human naive CD8 T cells. Elabscience human naive CD8 T cell selection kit helps you efficiently sort cell. Reagent preparation. Isolation buffer preparation. Filter the prepared buffer with 0.22 micrometer filter. Sample preparation and processing. Prepare human blood samples. PBMC sample is obtained from fresh human whole blood by density gradient centrifugation. For the density gradient centrifugation method of human PBMC samples, please refer to previous videos. Add the isolation buffer to the centrifuge tube. Mix by inverting. Centrifuge at 300 G for 5 minutes. Discard the supernatant. Add the isolation buffer to the centrifuge tube, and mix by pipetting. Transfer the cells to a 2 ml centrifuge tube. Centrifuge at 300 G for 5 minutes and discard the supernatant. Repeat the washing once. Resuspend the cells. Filter the PBMC through a 70 micrometer mesh nylon strainer and adjust the cell density to 1 × 10⁸ cells per milliliter for cell isolation. Cell isolation. Prepare 100 microliter of cell suspension. Add 50 microliter human naive CD8 T cell isolation cocktail. Gently pipette up and down 6 to 8 times with a pipette to mix. Incubate for 10 minutes at room temperature. Add isolation buffer to a final volume of 2 ml. Mix by inverting. Centrifuge at 300 G for 5 minutes and discard the supernatant. Resuspend the cells with 100 microliter isolation buffer. Place a clean flow cytometry tube or a centrifuge tube compatible with the magnetic rack into a tube rack. Pipette 1 ml of isolation buffer into the tube. Add 30 microliter of magnetic beads directly into the aforementioned 1 ml of isolation buffer. Mix by pipetting up and down 6 to 8 times. Place the flow cytometry tube or centrifuge tube on a magnetic rack. Magnetically separate at room temperature for 5 minutes. At this point, the magnetic beads are attracted to the tube wall. Keep the tube on the magnetic rack. Discard the supernatant. Remove the tube from the magnetic rack. Resuspend the magnetic beads using the cell suspension, and aspirate the cell suspension and pipette the beads off the tube wall to the bottom of the tube. Mix by pipetting up and down 6 to 8 times. Incubate at room temperature for 5 minutes. Add isolation buffer to a final volume of 2.5 ml. Mix gently with a pipette by blowing up and down for 6 to 8 times until no particles of magnetic beads are visible. Put the tube on a 5 ml magnetic rack. Stand for 5 minutes. Transfer the cell suspension to a clean centrifuge tube. This is the sorted human naive CD8 T cells.